So, good afternoon, everyone, and welcome to our first webinar of 2025. We are absolutely happy and pleased to have you all back with us as we kick off a new year of exchanging and sharing knowledge, education, and collaboration through our webinar series. And my name is Selma Kabashi, and I will be the host of today's session. Today we are honored to have an expert of the field, Dr. Lord Gowans, who will be sharing his insight into an important topic, especially for African, with the title of The Genetic Etiology of Syndromic Orofacial Cleft Among Africans. So Dr. Gowans holds a BSc in Biological Sciences and a PhD in Biotechnology from KNUST, Ghana, and the University of Iowa, USA, with advanced certifications in genetics and genomics from Stanford University, the Jackson Laboratory, and the University of Washington. He is a lecturer in the Department of Biochemistry and Biotechnology at KNUST. Dr. Gowans initiated Ghana's first genetics research on orofacial cleft and cranial sinustosis, employing cutting-edge genomic techniques to identify novel genetic susceptibility loci for these conditions. He established the Human Genetics and Genomics Laboratory at KNUST and collaborates globally, including on NIH-funded projects. With over 30 publications and 100 conference abstracts, Dr. Gowans has received accolades such as the NIH Emerging Global Leader Award and the 2023 IADR Smile Train Cleft Research Award. He is also involved in advancing ethical and societal aspects of genetic research in Africa. So before we start, I would like just to remind you that we do love your participation, and we encourage you, if you have a question, comment, or a point of discussion, to use the chat box or raise the hand function so we can address it immediately after the presentation. So without further delay, Dr. Lord Gowans, the floor is yours.
Okay. Good day, everyone. Thanks, Emma, for the nice introduction, and then good day to everyone. Happy New Year as well. I'm grateful for the organizers and thanks for having me. So as Sama rightly said, we want to discuss the genetic etiology of syndromic orofacial cleft among Africans. This is especially important given the fact that a lot of our countries do not have infrastructure to carry out genetic screening or tests as part of our routine medical care. So our research brings to fore some of the syndromes that are observed in association with clefting among Africans. So all the pictures I showed here that are not published papers, I have the ethical approval to share it from the parents of the affected individuals. So as we know, cleft is originate when the various structures of the face fail to fuse as we expect them to. So for example, when it comes to lip, when the median is a prominence and the maxillary fail to fuse, we have the lip and the palate result. For example, when the palatal shells fail to fuse in the case of the secondary palate, and then again for the primary palate when the intermaxillary segment fail to fuse. Now, when we go to OMIM, we have over 300 syndromes that may present with orofacial cleft. Taking clefting, for example, we know that we usually, for etiologic reasons, we separate them into cleft palate and cleft lip and cleft palate being the other group. For cleft palate, has a male to female ratio of 1:2, and it's estimated that as much as 50% of cleft palate is likely to be syndromic, compared to cleft lip and cleft palate, where we expect about 30% to be syndromic. And in that case, males are more affected than females. So most cases are obviously sporadic, with few being so sporadic non-familial, with few of them being familial. And the incidence, as we also know, vary by ethnicity, with Africans having as high as about 1 in 2,500 live births, and Native Americans and then Asians having the highest incidence. So here I show pictures of this a midline cleft lip, and that is macrosomia. That is a cleft lip and palate. That's unilateral to the right of the patient. That's bilateral. That's cleft palate. And that is an example of some of the facial clefts that we observe in our center. Okay, so we know the issues associated with clefting, the problems. I will not belabor this point. And again, the most important one to emphasize is the probable infanticide that may be associated with, and mortality that may be associated with clefting. And this is especially high when it's present with other congenital malformation in addition to the clefting. Okay, so just a quick view of the burden of cleft or syndromic cleft, for example, in our cohort using the paper we published. So, for example, in the paper, we recruited for about 2-year period, we recruited about 467 individuals. Of these individuals, actually just about 5% of them were multiple pedigrees. And based on clinical evaluation at a time, we were able to pinpoint about 12% of such cases were syndromic. Obviously, this diagnosis happened to be lower because at the time of evaluation, usually that they came in very young. So at the time of evaluation, we're likely to miss some of the syndrome because obviously some of the phenotype may not be obvious. Again, this pedigree usually are coming from low socioeconomic backgrounds. For example, with hoping over 60% or 70% living in rural areas. And again, the almost always coming from particular regions or particular portions of the nations, for example. So these are various, and there is a typical Van syndrome where we see the lipid, this typical Pierre-Robin sequence that we see. We also see individuals, for example, with hesodactyly. There's an individual with Apert syndrome, typical characteristic face of Apert syndrome, syndactyly of the fingers and toe. Then again, we have individual presenting with clefting and cleft foot, for example. So these are various syndromic forms that we see in our cohort. So as I said, so this is just a table giving us a summary showing that a lot of the cases, of course, in literature, it's reported that cleft palate has 2:1 female to male ratio, and that's what we see in our cohort. But when we come to cleft lip and cleft palate, usually it's almost 1:1 in our cohort, for example. So this is just a summary of the various syndromes. And again, as I said, based on the clinical evaluation, these are some of the syndromes that actually we could assign particular syndromes. Of course, for VUS, normally it's obvious when we see the lipid, we say that. And a lot of other syndrome. And for example, there were many syndromes. We could not assign any specific syndrome to it, and we classified them as morbid congenital anomalies because we lacked genetic services to help us to do that in our center. Now, in picking, we decided to pick a number of samples to carry out various studies that involve both syndromic and non-syndromic, and have come out with a number of syndromes that may be common in our cohort. So I will start by, for example, a large case study we did. This was actually a genome-wide association study that we carried out. But as part of those evaluation, we're expected to, for example, carry out gross chromosomal analysis. And during our analysis, we observed gross chromosomal aberrations that are associated with clefting. So, for example, in this paper, we show that there are individuals with clefting that may result from, for example, mosaic Turner syndrome. Some also resulting from XYY Klinefelter syndrome, for example. So all these are triple X syndrome as well. So indeed, we have instances of gross chromosomal abnormalities like changes in chromosome number. In this case, sex chromosome aneuploidies being associated with cleft. So these are some of the aneuploidies we observe. Again, apart from the sex chromosome aneuploidies, we also had the trisomies as well, trisomy 13 and trisomy 20. Obviously, trisomy 21 being the most common we encounter. So again, in this table, we show the age at which, for example, the mothers were recruited. And you could see that it is in line with literature where probably after 35 years, there's high likelihood of giving birth to a child with a trisomies. And one of the interesting things we observed was the fact that even some of the young folks that we recruited as controls actually had trisomy 21. But interestingly, because there was no screening, it couldn't be picked up. I'm sure they had to grow for a while before it could be picked up. So that is interesting. So we find this cross chromosome abnormalities as well. So both sex chromosome aneuploidies, like Turner syndrome, Klinefelter syndrome were observed, and then the trisomies as well. Again, apart from those changes in chromosome numbers, we also observe large-scale deletions and insertions and duplications, for example, that were associated with clefting. So these are a number of clefting that we show the chromosome numbers where we saw those kind of deletions and duplications. Of course, those kind of aberrations will involve large number of genes. And we also observe them in our cohort as well. So these are largely gross chromosomal aberrations that we observe in our cohort being associated with clefting. Okay, now apart from these large-scale chromosomal aberrations, which usually will not constitute large chunk of our cohort, we also have single gene disorders being associated with clefting. And as we all know, the most common syndromic form of clefting is the Van der Waals syndrome. And that syndrome is reported to account for about 2% of all clefting. So it's a very important syndrome. And I'm sure it serves as a model for even our understanding of non-syndromic clefting. So these pictures actually show very prominent typical Van der Waals syndrome where you see the clefting and then you see the typical lipid. Of course, these are very large lipids, so they are very obvious compared to the first one I showed where they were tiny lipids. And the second individual too is actually a Van der Waals, but actually you don't see the prominent lipid. Actually, you see just some dents as we see over here, 2 dents over here, but it's also a typical Van der Waals syndrome. So in our group, we've been able to about 3 different papers on this syndrome, trying to enumerate the kind of variations that are in this syndrome. So obviously, about 70% of individuals with Van der Waals syndromes will have mutations in the IRF6 gene, with some percentage also having a mutation in the grainy hair-like 3 gene as well. So those are those with cleft palate. So that will be about 5–10%. So still there are few instances where there are unknown genetic etiology, and obviously certain genes are involved. So in this paper, for example, what we did was to pick individual with Van der Waals syndrome together with others without the typical phenotype of Van der Waals. So they had cleft all right, but they were not lipids. So we picked them as well and included them, as well as other individuals with multiple congenital anomalies. And we did targeted gene sequencing of the IRF6 gene, looking for whether there are variations in them. So for the typical Van der Waals syndrome, yes, for our cohort, we're able to find etiology mutations in IRF6, and that confirmed the Van der Waals syndrome. But something interesting we also observed was that there were some of the patients that presented with just clefting without the typical lipid we see in Van der Waals syndrome. But interestingly, they also had variations in this gene, suggesting that they are potentially Van der Waals syndromes, just that the lipid phenotype wasn't penetrant. And that suggests that we could have many of such individuals probably coming to our clinic that may be Van der Waals, but actually the lipid phenotypes, as we could see, may be variable, as we see, or may not be penetrant in our population. And I'm sure it may not be something unique to our population, because elsewhere we've seen various lipids, variability, and even penetrance as well. So these are the different, again, as you can see here, all the variant we are classifying novel are variant that have not been reported elsewhere. So again, there may be, apart from variant that are shared, there could also be population-specific variant among Africans, which we must also take into consideration when we are doing this. So this is just looking at some pedigrees and the novel variant we observe among them. So these are, for example, two different families. One had non-syndromic cleft and palate. The other had non-syndromic cleft palate, meaning that they had just cleft without a lip phenotype. So we could see that there's variability in the clefting itself, but all of them had the variation, glutamate being substituted for lysine at 69 position of the protein, IRC protein, and that actually also occurred in both of them. Though the phenotypes are different, the families are not related. So that's one interesting observation. So you wonder what may be the kind of modification that is going on, that the expressivity is variable. So interestingly, in these families, we had other variation, for example, PAX7 in those individuals as well. And we asked whether, for example, PAX7 could modify the phenotype. So that's an interesting thing about Van der Waals syndrome we see in our population. Quite recently, our group also reported further cases of Van der Waals syndrome. And for example, novel variant were still observed. So that tells us that our cohort, our African cohort, is very good for novel variant discovery as well. Yes, we've been able to confirm some of the variant that have been reported elsewhere to also be etiology among Africans, but our studies continue to bring out new variants among Africans. Okay, so one interesting thing we also been seeing, for example, in our Ghanaian cohort, is the fact that Van der Waals syndrome, IR6 gene may also be involved in the familiar one. Of course, that's the gene underlying the VWS. But interestingly, so this is a pedigree of a family where there are 3 affected females. And if you look at the pedigree very well, the phenotype is variable. This individual is cleft and palate, that has cleft lip, and this individual has cleft palate. So sequencing of this family, for example, revealed a novel frame shift mutation that truncated protein actually in this family. Interestingly, the unaffected father also had this variant, suggesting the variant is not completely penetrant. So that is one thing we see about this family. And again, the variable expressivity of the phenotype is also interesting. As to exactly what may be modifying this phenotype in this family, or the penetrance of the gene, is something we look at. Looking at why all females will have the variant be affected by the males that are affected, raises a question whether sex could be a modifying factor in this family, for example. So this suggests to us there are families, whenever we see familiar cases where the lipids may not be visible, probably this IRF gene may also contribute to that. Now, this is not actually the only family. For example, my graduate student, Bruce, is working on about 25 multiplex families from Ghana and trying to look at the variations responsible for this. So we show here 2 of such family, for example, where again, this having CP, the other one having CP as well. And he shows variant in 2 genes that are associated with Van der Waals syndrome. Obviously, these families too, we don't see the lipid phenotype in these families, but the cleft happen to be familiar in this family, the mother transferring the phenotype to the affected daughter. and then the proband inheriting from the mom in the other family. And in the first family, we see variant in IR6 gene, and in the other family, we find variant gray hair like 3, which we said that is accounting for VW vulnerable cases in about 5 to 10% of the cases, particularly those cases that present with cleft palate, and that's exactly what we are seeing over here. So that gene actually is also involved in familiar cases in clefting as well and among our cohort, where, for example, there are no lipids, but still when we see familiar cases, that tells us that these genes could play a very important role. Of course, it's not only the gene we are seeing other genes as well, but for the sake of the syndromes, I'll concentrate on those. Okay, so one of the cohort we also see in our center, for example, is cleft presenting with club food, for example. So in this paper, we had a cohort of 6 individuals. At the time of evaluating them clinically, the only phenotype we observed were actually cleft lip and palate, for example, NO palate, and then the limb abnormality, which was happy to the club foot. So initially, we had hypothesized, for example, that probably there is a common underlying genetic etiology. So we carried out the whole exome sequencing for this group. And interestingly, observations suggested otherwise. So they look clinically the same or very similar as far as the phenotypes are concerned. But actually, we saw that different genetic syndromes actually underlying them. And that was actually an interesting observation for us to know. So, for example, in this family, we see that there's 3L associated with Perlman syndrome. We also saw Vicky syndrome, Fraser syndrome, and Treacher-Collins syndrome, and one, for example, having the Charge syndrome and then the cardiomyopathy. So, as I said, of course, there were other phenotypes probably might have missed because these children really present very early to our clinics. Unfortunately, once the cleft is repaired, you hardly see them coming back unless some few follow-ups. But once it's over 1 year, they don't come up. And unfortunately, as I said, the syndromic ones are also prone to mortality. Because of that, a lot of them even don't come back for the repair. So because of that, we don't have the luxury of getting to know all the phenotypes that may be associated with it. But obviously, these observation from genetic evaluation suggest to us that there could have been additional phenotype that we missed because of the time of evaluation. Okay, so recently, our group also decided to carry out about a large cohort of syndromic individuals, and what we are trying to do was to decipher the etiology of various syndromic forms of clefting. So we sequence about 50 families with different forms of syndromes. And one of the syndrome that was very, let me say, sequence that was very prominent in our center is a Pierre Robin sequence. So this is my graduate student, Christian, and he's working on Pierre Robin sequence. He has about 18 families with Pierre Robin, which we did whole exome sequencing at deep sequencing, of course, at 100x. And we know the typical features of Pierre Robin sequence, the micronathia, and then the cleft palate and prosoptosis. So all of our individuals at least had these 3 phenotypes, and this is a Pierre Robin individual from our center. So as I said, our aim was to characterize variant that may be responsible for Pierre Robin sequence in our cohort. So we collect these samples from these families. We carry out whole exome sequencing at 100x, that's deep sequencing. And we use the Sentinel pipeline, then guided by the ACMG evaluations, employing population allele frequencies, employing variant effect predictors, employing databases, segregation analysis. As far as ClinVar and others, we're able to evaluate the sequences and to look for probable syndromes that may be responsible for PRS in our cohort. So this is just some of the variant that we observe in our cohort. So you could see, we see the typical collagen genes. And in fact, that actually top the collagen and collagen-related genes were the major ones that was a greater percentage of our cohort, suggesting largely in line with literature has that Stickler syndrome may be a major contributor to Pierre-Robin sequence in our center. And one of the interesting genes, which is also not been well characterized as far as Pierre-Robin sequence is concerned, is PRDM. And in our cohort, we find novel mutation, that frameshift mutation, in two different families. The same mutation have this PRDM gene. And as I said, this gene has not been well characterized, and we may need further functional study in that. So when you pick Pierre-Robin sequence literature, I'm not sure you are going to find this gene, but our evaluation suggests this may be an important gene for this kind of syndrome in our cohort. Again, so the data I'm presenting now are about publishing such data. And for example, we also find an interesting de novo mutation, a mutation in SAT-B2 gene. And of course, the CREP-BP, which is well known for Robinson-Terry syndrome, we also find variation in this gene in one of the families. So of all the genes we evaluated, we picked, for example, our interest was to look at which molecular pathways, gene function they were involved. So this gene ontology, using the G profiler, we did this analysis, for example. And in the molecular pathway, we see, for example, that these genes are involved in extracellular matrix structural constituent. And that is very important. For example, we see collagen trimmer, extracellular matrix. Of course, all these are important for connective tissue formation. So it's interesting to show that our genes are involved that part, those pathway, and that actually support the probable way function in the abnormal mandible morphology that we see. And of course, Kirk pathway, reactome, all of them are bringing up that our genes are involved in interesting pathways that are involved in, for example, formation of the extracellular matrix. Again, the human phenotype ontology also agrees that this gene actually, for example, involved in abnormal mandible morphology. And then again, we also did further evaluation, for example, by importing this into cytoscape and look at the various pathways that actually interact for this. And again, we see that this pathway indeed, the genes are in pathway associated with, for example, abnormal mandible morphology and telophase equinovarus, which is cleft food, for example. Again, this is just a strange analysis trying to look at the interactions between these genes that we observe in our cohort, and we could see that there is actually a network of between some of them, as we see over here, and then some of them being novel. And we also use the cytoscape to look for the hub genes within this. Of course, we see the CREP-BP being a critical gene in the network of genes that involve in Pierre-Robin sequence. And we also see the collagen genes as well as CD36, for example, being cardinal genes in this pathway. So in SS, for our evaluation of 17 families, we see various syndromes associated with Pierre-Robin sequence. So as Riley calls the sequence, because we expect there are many syndromes underlying it. So in this case, we see that Stickler syndrome. So we find mutations in the collagen genes, SAT2B, which is collagen related. So the Stickler syndrome happens to be the most prominent syndrome. And of course, the PRDM, which need further characterization, also observed in a number of families. And one family with Robinson-Terry syndrome. And we have other interesting genes that are associated with not well known, for example, the Criducat syndrome from safety NDD2, for example, and many other of these syndromes. Now, one of the interesting things is, I think that 3 of the families, we find pathogenic variation in ABCA4. Now, this ABCA4 has been associated with cleftin, a genome-wide significance, but we actually don't know it's associated with any cleftin syndrome. But it was interesting to see about 3 different families having etiologic variation in this gene, and that variation also is classified pathogenic by Clinvar. So probably this may be a new syndrome that we are seeing being as PRS syndrome. for which ABCA4 may be a major player in the syndrome. The other thing we found in our cohort is the OFC occurring with other limb abnormalities. So this is Edna. Edna is working on OFC present with limb abnormalities, including whether hand or other abnormalities. So recruit family, the same thing, look at inheritance pattern. And one of the interesting things is the syndromes we identify. So for example, one of the syndrome, clinically, it was said that the syndrome happened to be Carpenter syndrome because what was observed. But actually, after evaluation, the other evaluation, we saw that that is actually an APES syndrome, for example. So her evaluation brought up a number of syndromes that are associated with limb abnormalities and clefting. We have Cornelia de Lange, Freeman-Sheldon, APES syndrome, EC3, and then other syndrome, for example. Again, some of the genes showed pleiotropic. In fact, that one gene showing two different, being responsible for more than one syndrome. And again, some of the individuals had a number of variations. So this is just quick overview of the families she has, and then the phenotype they present with. And evaluation, interestingly, this cohort, a lot of the variation we see with clefting and then limb abnormalities happen to be due to de novo variation, de novo mutation in individuals. So apart from the family that had the Cornelia de Lange syndrome, that had novel deletion in NIPB, which is a cardinal gene for that syndrome. And then there's also another family that has EEC3, ectodermal dysplasia syndrome 3, which we did not get the fathers for which we couldn't determine the de novo. Interesting, all the families actually have de novo variations in them. And some of them well-known genes, others not well-known genes. So for example, we see the farm group of genes being represented in our cohort. For example, farm 70 being responsible for the vector syndrome, for example, and other farms showing up in our syndrome as de novo. Again, we find de novo mutation, for example, the Freeman-Sheldon syndrome in myosavage gene 3. So it looks like the limb abnormalities together with the clefting are usually due to de novo variations in them. So this is one of the families where we had a TP63 variation. The mother, for example, presented with the ectrodactyly, but the mom did not have any clefting phenotype. And he also had the variation that we saw in TP63. But the child had ectrodactyly, as we see here, together with cleft and palate. So again, here, the mother has a phenotype, has a variation, but again, the specificity of the various phenotype was actually variable in the family. Okay, so this is just modeling of so again, when you look at her enrichment analysis, looking at the pathways involved, using various modeling tools, and then looking at conservation, we see epithelial cell differentiation, we see appendage morphogenesis, we see skeletal system development, cranial skeletal development, and embryonic morphogenesis. So that suggests that the genes we find in this cohort actually involve in pathways that make biological sense in the context of cleft lip and palate, or facial cleft, and then the limb abnormalities we observe. Now, another cohort we have is craniofacial microsomia, or golden hair syndrome, or hemifacial microsomia. I'm sure this syndrome has a lot of names, and Rahel is working on this syndrome. So interestingly, over here, I list some of the genes that have been associated with this syndrome. Of course, the genetic study on this syndrome looks to be in its infancy. And these are some of the genes that have been implicated. Interestingly, we have about 7 or such families that we're evaluating. Whole exome sequencing did not observe any of these genes, apart from the NLRP, where we find a mutation in that, a variation in that gene. But interestingly, that variant was also in the mom, though it's presented to be pathogenic, so it was inherited. Now, these are the various variants that we find in our cohort. So I'll highlight our top one, for example, RANX1. We find, interestingly, one of the variants that also came up, interestingly, is the PAX6. And this PAX6 variant was found in different individuals. Now, if you go to Genome Alliance, it's shown that the PAX6 actually exhibits some of the phenotype we see in typical craniofacial microsomia. And these are the other genes. As I said, these genes are not typically what have been reported in literature to be associated with craniofacial microsomia, suggesting we may have different genetic etiology when it comes to our population, for example. So these are the other genes. And again, the network try to bring out the kind of variation or gene, the network over here as well. Because of time, I'm able to expand further on that. So these are various genes. Again, for example, one of the genes you observed was the HKDC1, which has been associated with cleft and genome-wide significance. And over here, she reports novel variant in that gene are being associated with craniofacial microsomia or golden heart syndrome or hemifacial microsomia. Okay, so those are the various variants that we observed in our cohort. And as I said, it looks interesting. And one of the things that we see that if you pick any one particular family, you may have many interesting genes. And as I said, we may have to do some functional experiment in order to validate some of these variations, especially, for example, PAX is well, the PAX group, the paired box, Hobie box group, are well known for clefting. And for example, dental anomalies, we know PAX7 is well known clefting gene. So for PAX6 to pop up here also looks interesting to us to be associated with craniofacial microsomia. And as I said, again, to find the same frameshift mutation in two different families also interesting, and that required that we do some functional study to validate these kind of observations. Of course, there are other results, and we are also looking at on facial clefting, for example, and I'm sure we get a result up very soon. Okay, so lastly, it's about, again, a paper we publish. So this is a consortium, not just from our center. A group contacted us doing similar work, and we find variations in AMOT-L1 gene. And now this gene, it looks like the variations were actually concentrated in a particular hotspot. And the individual had the kind of phenotype that we are describing over in this table. And for example, the cardinal phenotypes being orofacial clefting, cardiac abnormalities, and tall stature, for example. And all these were due to variations that were concentrated in a particular domain of the AMOT1 protein, for example. Now, so these are the various syndromes. So in our cohort then, we are seeing both gross chromosomal syndrome associated with gross chromosomal abnormalities, as well as syndrome that are due to single gene disorders. Okay, so to conclude, we know that both chromosomal and single gene disorders are responsible for the syndromic cases that we are seeing in our cohort. But one interesting thing that we are seeing is of the variable penetrance of the phenotype of the variant. So for example, in the case of the IRF6 gene, where we are having variable penetrance even in multiplex families, and not even in multiple, even in sporadic cases as well, where the penetrance may not be, may be found, some variant may be found in unaffected individuals, and then may be found in the affected individual as well, suggesting there may be some genetic modifier responsible for that. And the pedigree I showed earlier on suggests that probably sex may be a reason for that. Again, it's about the variable expressivity of the phenotype we are seeing. We saw the EEC3 family where they all have the same mutation. The mom has just the ectrodactyly, but the child had the ectrodactyly and the cleft lip and palate as well. So we were, again, we're looking at the same syndrome, but actually the expressing may vary. I also talked about the lipid phenotype, for example, in the case of Van der Waals syndrome, where there may be good, the lipid may be extremely large, intermediate, or there are instances you may see just an indent, or in some cases, as we see in some families, there may not be no lipid at all, but actually then you still have the individual or the family being a VWS individual. Again, as we said, similar phenotype in the case of Pierre-Robin sequence, they may suggest the cleft palate and of course the micronathia, but as literature rightly suggests, it may be different syndrome. Yes, Stickler syndrome happens to be the most prominent in our cohort, but it looks like there are many other syndromes that may present with the cleft palate and the micronathia as well. And there were instances also we see the same phenotype, but the underlying etiology is different, as I demonstrated in the case of cleft presenting with limb abnormalities, specifically the case of cleft presenting with club foot, for example, where we had envisaged a common genetic syndrome, but it happened that there were different underlying genetic syndromes. Again, we saw that Van der Waals syndrome probably not presenting or not presenting with VWS may be a very interesting syndrome in our cohort when it comes to familiar cases, in a sense that both genes that are implicated in Van der Waals syndrome, being IRF6, and grainy hair-like 3 genes, can all appear in familiar cases. The responsive familiar cases where we don't see even any lipids phenotype. Again, we talk about the Stickler syndrome and then the PRDM9 genes playing cardinal role in the Pierre-Robin sequence. Again, when we talk about the cleft lip occurring with limb abnormalities, we showed again there are different syndromes that may be underlying that. Now, our studies has many implications, of course, for application of clinical genetics in our setting, in a sense that there may be many variants that are yet to be discovered, even when it comes to well-known genes like IRF6, because as I demonstrate in our call, we find many novel variations that have not been reported elsewhere. Now, apart from that, as I'm demonstrating through, for example, Pierre-Robin sequence cohort, when it comes to, I'm sure, generating gene panels, probably in the future for syndromic clefting, we may have to consider African-specific context genetic architecture when it comes to developing these kind of gene panels, in that we may pick up from other population, but these may not be genes that may be responsible for the ones in our cases. And this does not only apply to clefting. I'm sure I'm also working on cranial sinustosis, and the few that we sequence, of course, we find mutation in FGFR2. But for example, apart from FGFR2 variations, all other individuals, we have about 16 different families. None of them have the mutation in cardinal genes like FGF3, FGFR3, and many others that are well known. So these have implication for us as Africans when we want to generate gene panels that we may use to screen craniofacial abnormalities. So I acknowledge the contribution of my mentors. I acknowledge all references I use here. Very grateful to family that participated. Very grateful to my mentors, both in Africa and elsewhere, Prof. Butali, Prof. Peter Duncan, Murray, Prof. Murray, and all those who contribute to this. Not forgetting those who are funding our research, the National Institute of NIDCR, Fogarty, Ghana Clear Foundation, of course, IADL and Smart Train as well. Very grateful to all of them. And grateful to these are my members of my lab, my graduate students who are doing very well when it comes to this. And then thank you. I think I have to leave some time for questions for us to interact. Thank you very much for your attention. I'll be glad to have questions and your comment.
Thank you very much, Dr. Gowans, for this wonderful presentation. I can see lots of novelty in your work, and it really addresses an important issue, critical issue, basically, in Africa. Let me see. I see. I'm looking if there is a question. I see Prof. Butali shared an article. Lots of clapping. I had a quick look at that article if you would like to comment on it. Oh, I have Prof. Botali. Prof? Oh.
Yeah, thank you very much. And thanks to Dr. Gowans, very well delivered lecture and very insightful. So Dr. Gowans is a demonstration of capacity building, knowledge transfer, and the future of craniofacial research in Africa. So I just shared the article on the functional work on PRDM 16, and I would like your student to take a look at that. That will give you an idea of what has been done for PRDM 16 and PRS-like phenotypes. Otherwise, I think you're doing a good job, and I like the fact that you're passing on the baton to the future beneficial geneticists on the continent. Thank you.
Thank you, Prof.
Yeah, Prof.
Just to add, so Prof. Botali happened to be my PhD supervisor when I was in Iowa. So he, together with Prof. Peter Donko, Prof. Mari, they mentored me in Iowa. So I'm grateful to them.
That's wonderful. There's another hand raised. Bruce, you may unmute yourself.
Sorry, I have a question. I agree this is a wonderful work and contribution to capacity building and all these great works to junior people in Africa and elsewhere. So my question to you is that you see the same phenotype, multiple variability in terms of expressivity and other things. Have you done some gene environment interaction? Is there any specific environment for some of this variation that you can account to?
Okay, so that's something we've not done yet. Looking at the gene environment interaction in these instances. I think for the non-syndromics, I think we've done that once. I think for one of our publications, we tried to look at. I think for my PhD team, that's what we try to look at for gene environment interaction. So usually for the non-syndromics, for example, environmental factors like folate deficiency or any other factor that may affect folate metabolism may be very important. So we've done that. But largely for syndromes, of course, we don't discount the role of environmental risk factors, all right. But largely for syndromes, we expect a lot of them to have the Mendelian, for example, in nature, for which reason largely they will be genetic. So yes, for the syndromes, we are yet to do any gene environment interaction for that. But again, we don't discount the possibility that environmental risk factors may modify the phenotype, for which reason we see variability in the expressivity.
Yeah, thank you. Thank you very much.
Dr. Gowans, I understand that most of your work have been done in among Ghana population. Is that correct?
Yes, so that is for the syndromes that I presented, most of them here. But obviously, no, I work. So when it comes to, for example, the Van der Waals syndromes, we've had samples from Nigeria and Ethiopia as well. So Prof. Botali directs the African Craniofacial Anomalies Network. So we have collaborators in Nigeria, collaborators in Ghana, collaborators in Ethiopia, and even South Africa. So we work together on these projects.
Okay, and the genes or gene mutation were similar in most of those, like ethnic-wise or population-based. Did you see any differences?
Okay, so unfortunately, for example, the PRS, the limb abnormalities, and then the craniofacial microsomia, I'm presenting all these are from Ghanaian cohorts. So I don't have a sample from the other populations to look at it. But for when it comes to the Van der Waals syndrome, we've sequenced sample from Nigeria, from Ethiopia, from Ghana as well. For which reason, for example, we've seen some of the variant being shared among us, and then others being specific, for example. So let me go, if my memory serves me right, for example, this work that I did as part of my PhD over here. So this Van der Waals syndromes, for example, some of the variant we observe in both population. So for example, the glutamine variant at position 69, that variant was observed among Ghanaians, for example. However, the 65 was observed among Nigerians. So obviously there may be certain variation that may be specific to certain populations. But again, there's some of the variant too we share that we see them in both cohort, because some of the variant have been reported. We saw them in Nigerian population, we saw them in Ghanaian population as well. However, some of the variant too are maybe population specific. And that is expected because, of course, our PCR analysis during GWAS suggests that we are not homogeneous population. So obviously we expect certain variations when it comes to say genetic polymorphisms.
So I think that will be a great opportunity to suggest for the audience of anyone from other African countries is conducting or going through similar project in other syndromes than Van der Waals. It would be nice to collaborate and share the results to compare.
Obviously, it would be great to do that. And I think our network, Director Batal, is open to collaboration. And that is what we intend to do. We want to tell the African story. So our arms are open, and we hope to collaborate with as many Africans as possible so that the story will be representative of Africans. And that's exactly our aim. So going forward, I think we can contact any of us, and we are happy to work together.
That would be great. I don't see any other questions.
I have minor question. Probably I don't know the phenotype well. Are you considering this as monogenic inheritance, or do they have this polygenic as well?
Okay, so for Van der Waals, for example, in this instance, a Mendelian one, so it's monogenic, but it's monogenic, all right, but it's not always emanating from a particular gene. So as I said, for example, IR6 will account for about 70%, and then grainy hair like 3 about 3%, and then other variation as well. I'm so very sure you will see a publication from our group bringing out other genes as well. So yes, it's monogenic. So a variant in RF6, for example, is both sufficient to cause a variation without any other contribution from any other gene. So in that instance, then it becomes monogenic. But cleft in general, we cannot discount that, for example, the non-syndromic forms are not polygenic. But for the syndromic, for example, the VWS, and then a lot of the syndromes that I presented, largely, I'm sure they are monogenic in nature.
Did that?
Thank you.
Okay, it seems it answered your question. So any other questions? I don't see any other raised hand. Nothing in the chat box. So I think we're coming to the end of our webinar.
So if I have a minute, I can ask one more.
Yes, please. Yeah.
Sorry, I just want to learn what is the future direction you are proposing here. Probably I missed that point. What's the next step?
Okay, so the next step, as I said, I'm sure these are interesting discoveries, but as I said, one of the things we hope to do is to extend our tentacles to get other African countries on board, for example, so that we can the story representative of Africa. And as I said, that may influence, for example, when it comes to having gene panels that we can use to screen our syndromic forms. I'm sure that this is one of the things that it can influence a lot. And again, this can also influence our clinics, as I rightly said. I'm sure apart from South Africa, that has well-developed genomic infrastructure. Many African, Nigeria is coming out, and probably Ghana and other, but largely Africa is underrepresented when it comes to such infrastructure. So I'm sure these observations will also influence clinical evaluation of the phenotypes that present in our clinic. So in the long run, as I said, we intend that this should influence, for example, gene panels that may be developed for our population, and also extend the collaboration to include other African countries, so that when we are developing set panels, it will be representative of our people on the continent.
Very good. I was also hoping, if you are planning to do some, besides variant identification, if you have other additional omics type, or environmental or lifestyle, or some other additional work besides gene panel.
Okay, so yes, in our cohort, yes, we also collect environmental data. We just don't collect the DNA, we collect environmental data as well. But we are yet to analyze those ones. So all those information are collected as well. And then apart from the gene discovery, we also have collaborators. So for example, during my PhD, I worked on zebrafish, using the zebrafish as a model to study clefting, and I still do that. So that is some of the things that I was saying, that we have to use the animal models, for example, to test the new genes that we are discovering in our cohort. So yes, we have collaborators around, and we do functional genomic studies using the zebrafish models and mouse models, for example, to validate some of the genes and variant that we observe in our cohort. So yes, those omics studies, we extend it beyond the genomics to involve the functional genomics as well.
Wonderful. Thank you very much.
There's a question in the chat box, Lord. Variations among various populations potentially impact phenotypes of severe syndrome. How has this impacted your ability to identify various syndromes in your study population? Should we expect phenotyping of various syndromes to be population specific?
So this is an interesting one. So I'm sure if you pick, let's pick something like Down syndrome. I think when we're coming up and reading literature, there were instances people thought that probably Down syndrome did not exist among Africans, because probably how they presented, they look like among Africans. So I'm sure for if you come to craniofacial phenotypes, we know that African faces, for example, the facial profile of Africans look a bit different from that of Caucasians. And even within Africa, for example, we know that the faces of Ethiopians and then somewhere in East Africa look similar, like the Caucasian face, compared to those of us in West Africa, for example, West and Central Africa. So obviously, the population that you are dealing with may also influence your ability to pick up certain phenotype and all those. So for example, one of the things that one of my graduate students was doing, for example, was looking at facial variation among families affected with cleft and those not affected. And indeed, we are finding, for example, that certain variations are common in families with cleft and those without clefting. There are differences in that context. So yes, I don't discount the fact that population differences, especially when it comes to craniofacial phenotype, because of the variations in our physiognomy, we are likely to miss certain phenotypes, that is craniofacial phenotypes, if we we use particular metrics for all family or all populations, even within Africa, because of the variations in our facial profile. So I'm sure if you pick a typical West African face and then compare to a typical East African face, you're going to see a lot of variation. For example, the broad face we see it among West Africans compared to that the relatively narrower we see probably in East Africans, for example. So obviously, yes, population differences were going to affect our ability to pick up craniofacial phenotype. But I'm sure the genetics may largely be the same among us, but our physiognomy may influence how we pick our morphometrics, may influence our ability to pick up some of these. And again, I'm sure one of the things we are lacking is we don't have trained dysmorphologists in most of our clinics. Dysmorphologists trained in the African context who know problem variations in our facial profile, for example. We don't have them in our clinics, so it's obvious there are certain things we may mix. If they are not very obvious in our population, then we are going to mix those ones. So that would be my take on that submission. Thank you.
Yeah, that was a great question with a great answer. And I see that, Bruce, you have a raised hand. Do you have another question? Okay. I think our time is up for today. And thank you again very much, Dr. Gowans, for this enlightening presentation. And to all our attendees, we appreciate your time and your thoughtful questions. Just before we close, I would like to extend an invitation to our community. If any one of the attendees would like to present in the future in our webinar series or wants to nominate someone that has an interesting topic, please go ahead and you can pop us an email. I will write it down in the chat. And final reminder that the recordings of today's webinar will be available in our website in just the upcoming few days. So thank you again for joining us today and have a great day further. Thank you very much, Lord.
Thank you for having me.